The overall goal of this procedure is to measure oxidative stress resistance of Caenorhabditis elegans nematodes in liquid. This is accomplished by first dissolving ROS-inducing agents such as paraquat in M9 buffer and aliquoting the solution in a 96-well microtiter plate. The second step is to transfer 5 to 8 synchronized late L4 nematodes to every well, using at least 12 wells per condition. Next, using a dissecting microscope, the number of living and dead animals is determined every hour until most animals are dead. The final step is to calculate the percentage of animals that survived at every time point for every condition. At least 6 days prior to performing the oxidative stress resistance assay, prepare the modified Youngren's only bactopeptone or MYOB dry mix for making the MYOB medium. Mix well with shaking. To prepare 1 liter of MYOB medium, combine 6 grams of the MYOB dry mix with 17 grams of agar and make up to 1 liter with H2O. Autoclave for 45 minutes at 122 degrees Celsius. Pour the MYOB medium into plates and let the plates dry at room temperature for 2 days. Using sterile techniques, inoculate 100 milliliters of LB broth medium with E. coli OP50 bacteria and grow overnight at 37 degrees Celsius.